Hi everyone! In today's video, we'll be talking about cyclic peptides, and we'll see how they, as a class of drugs, will tide us through a new era of drug discovery. And before we start, I would just like to say hi, I'm Odilia. In biomedical and its related fields of pharmaceutical research, there is, and will always remain, an ever-growing need to create new drugs. This is because old drugs have not been able to adequately tackle old diseases, or even because new diseases have arisen. In this new era, the focus of drug discovery has been on cyclic peptides. Cyclic peptides are simply amino acid sequences, and an amino acid is a molecule made up of an amino group, a carboxyl group, and an organic R group that is unique to each amino acid. When the carboxylic acid group of one amino acid approaches the amino group of the second amino acid, a peptide bond can be formed between them. In this manner, when multiple amino acids are bonded together through peptide bonds, an amino acid sequence is formed. But what makes a cyclic peptide different from a non-cyclic one is that cyclic peptides have a loop structure within the peptide chain. In the pharmaceutical industry, cyclic peptides have shown to be very successful as drugs. They have shown to effectively target diseases and are widely applicable to a whole range of diseases. Cyclic peptide drugs can serve as antibiotics, antibacterial and fungal drugs, and immunosuppressive drugs, amongst others. There are several favorable properties that allow cyclic peptides to be so successful as drugs. The first property is their display of high selectivity for protein targets. This is because cyclic peptides are moderately large, which allows for a large contact area with the protein target. At the same time, diverse functional groups that are present on the peptide allow for high selectivity as well. The second property is their high affinity for protein targets. The strong binding ability of cyclic peptides comes from its large surface area, along with the peptide's ability to form multiple hydrogen bonds. The third property that cyclic peptides possess is good serum stability. Enzymes in the blood are hindered from degrading the peptide. This is because these peptides are cyclized and do not contain ends that can be easily cleaved by enzymes. The fourth favorable property of using cyclic peptides as drugs is their low pharmacotoxicity. Low pharmacotoxicity refers to low exposure to any toxicity that might arise as a side effect of inducing cyclic peptides as a drug. This ensures that even if the cyclic peptides were to undergo enzymatic degradation, the products produced are non-toxic and does not cause poisoning to the patient. Due to the biomedical potential that cyclic peptides possess, they have made major contributions to the pharmaceutical industry. But maybe you aren't feeling too convinced yet. So, let me convince you with some prominent examples of cyclic peptides that are being used as drugs. One prominent example is cyclosporin A. Cyclosporin A is a prominent immunosuppressant isolated from a fungus, Tolypocladium inflatum. It is used clinically worldwide to prevent graft rejection. This means that those who undergo rheumatoid arthritis, Crohn's disease, and organ transplants have this drug prescribed. In the USA alone, the annual sale of this drug is more than $1 billion. Say what? $1 billion? Yup, you heard that right. That's how prominent this cyclic peptide drug is. Another prominent cyclic peptide drug is Vacazole B, which is an anti-tumor drug. It is a cyclodecapeptide, meaning it contains 10 peptides in a cyclic structure. It was isolated from strains of a cyanobacteria, Muria producens, which was collected from the Red Sea. Vacazole B exhibits potent cytotoxic activities against human lung cancer cells and human breast cancer cells. Oh yeah, leave us alone, cancer! You might not have heard about the two names that I just threw at you, but maybe you've heard of this medicine called tyrosol. It is a commercialized antibiotic which is being used in clinics today. It contains the cyclopeptide tyrosidine. Furosemide is a cyclodecapeptide, just like Wakazobi, which again means it contains 10 peptides in a cyclic form. It was found from a culture extract of soy bacillus, Bacillus brevis, as early as 1939. What is significant about it is that it was found to have good antibacterial activity. Isn't that great? Not just the last example, but all these examples of cyclic peptide drugs and how they can be used. Over 40 cyclic peptide drugs are currently in clinical use. And on average, around one new cyclic peptide drug enters the market every year. Right now, over 20 cyclic peptides are currently undergoing clinical evaluation. These molecules have been developed for a highly diverse set of medical conditions. Some of these include several types of cancer, various infectious diseases, endocrine and metabolic disorders, and cardiovascular disorders. You might have noticed that in all the examples, the cyclic peptide compounds were all obtained from nature. It is true that the vast majority of these cyclic peptides are derivatives of natural products from organisms or human hormones. But now, with new and powerful techniques, synthetic cyclic peptide drugs can also be easily developed for targets. Synthetically made products complement the situation in which natural products do not offer solutions. In this way, we can introduce fresh drugs into the market. Drugs that are possibly more effective than the ones we currently have. Drugs which may provide the solutions for both old and new diseases. And drugs that will tide us through a new era.